a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells, because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your to the ends. Kind of helps them move along.
Making it into 46 chromosomes on this side and 46 on this side. Now, once this is done, when they are pried apart towards the end of that entire cell, telophase is when the nuclear envelope starts developing around these two. Okay, now the cell is ready to completely divide into two different cells, and that is what is achieved in cytokinesis, where one cell gives rise to two daughter cells with the same number of chromosomes, same function, and same everything. Okay, so this is how usually the function, or basically the entire process of mitosis, takes place. But what is meiosis? Meiosis is a little different than mitosis. We'll not see the video, that you can see later. But now, just like in the stage of mitosis, it will replicate, because you need to make sure that you are dividing. Now, when it comes to replication, there is a very important process that happens here. Okay, it is called recombination. Okay, I'll just show it to you. So when it comes to the process of meiosis, there is an interphase. Okay, here the homologous chromosomes, what does homologous mean? They are similar in structure, okay, and they look similar alike. What they do is they if these two are the chromosomes, they kind of exchange information during this particular period. Why does that happen? Have you ever wondered why, if you and your sibling come from the same parents, there's so much difference between you two? That is what is achieved through meiosis, because during this time, the mother and the father chromosome sets, when they recombine, there is an exchange of information which gives rise to variation, which is the reason why it leads to uniqueness. Okay, so while mitosis is going to focus on your growth and cell repair, etc., meiosis is going to be the result why all of us are so unique in our own right. Okay, so meiosis is called reduction cell division. Mitosis in this particular process is one of them being chromatin accessibility. Do you know what is chromatin? Now, when your DNA, before it gets condensed into chromosomes, you have your chromatin. If you remember, DNA, if it's coiled around those histones, it looks like beads on a string, right? Now, this particular string in between, what if it is relaxed? Don't you think it's open to receiving, it's open to being translated very easily? Now, if I hold my hand like this, I'm holding a message and I tell you, why don't you read it? And you'd be like, I can't see it. You need to open it. So if I open it, it is going to be ready for translating. Similarly, your chromatin fiber that holds the entire DNA, that beads on the string structure. Now, if your DNA, it is tightly wound around a histone, it is not ready for it to be open to, transcription and then further translation. So in cases where a particular stretch of DNA is open for transcription on that beads in a string structure, your mRNA will easily be able to read it, and then that is going to be translated. So chromatin accessibility is responsible. Sometimes it is not read. The protein that is to be made, if the message or the codons are not being read properly, how is it going to make? So chromatin accessibility, the accessibility to the chromatin fiber in order to make a protein sometimes is responsible. Okay, transcription is the key factor, right, in order to for the translation to happen. But even during this time, what happens is certain transcription factor proteins, when they bind to specific DNA sequences or on a gene, they can either promote or repress its transcription into RNA. So whether it is binding to the promoter region, okay, or the termination zone, it depends on whether or not that particular gene would be transcripted or that particular stretch of DNA would be translated into a protein. Then you have RNA processing. Do you remember when I told you before mRNA leaves the nucleus and towards the end, even when the protein is processed, during this processing of RNA, sometimes the RNA could get chopped up, or splicing, capping, or any other process basically that could render the mRNA unreadable. Okay, so sometimes when before the mRNA leaves the nucleus, if there is an issue in that area, of course the mRNA won't be able to carry the necessary information to be made. Okay, and also there is something called small miRNAs. Okay, so these are also precursors, but they can also cause an issue wherein the actual message does not translate or it does not reach, or that leads to destabilization of your actual mRNA. Okay, so these are all the processes, or these are all the issues that could arise that could hinder the translation process or the transcription process itself. Okay, during translation, what could happen? The translation of mRNA may be increased or inhibited by regulators. What regulators? Your own genetic regulators. Okay, protein activity. Now, before the final protein is made, during the process of folding and processing, it could also be interfered by external factors. Okay, so now that could also change the activity or the behavior or the function of the protein. Okay, sometimes when your mRNA is carrying the information, let's just say there is a viral RNA also that comes in. Okay, that also can very much hinder or interfere in the process. Okay, so this usually are the cases where the gene regulation or expression can get hindered. In the end, what is gene expression? A protein is made, which is the reason why you saw all of those factors that could hinder gene expression. Okay, so this is a very short chapter. It is not much to look into, but it is important for you to understand what could be the hindering factors towards gene regulation. And why gene regulation is important is because that is what decides finally the roles and the functions in our body, and that also gives us the power to actually correct our factors that we can modify and control to change the gene expression. We also have the power to down regulate or upregulate certain genes. Okay, so this is what gene expression and regulation is all about. Okay, let me play a short video for mutations and their types, so you will understand what that is. But before we go into the video, mutation, okay, what do you understand by mutation? Let me just get answers from you before we go into the video so that, we'll have a little exposure. What do you think mutation is? Thank you so much, Sumit sir. What exactly do you understand by mutation?
So to make it a little more interactive and keep asking questions, get into a discussion.
We are really brilliant, students in this batch. And honestly, what I have seen is, they're very interactive in any case when it comes to answering. But even if you don't have any questions, you can share whatever you want, or, something that you know about this particular subject. So, what, even if others don't know about it, they could hear it from you. So it could be interactive even in that manner. So if at all you want to share something that you have experienced in these chapters, or something that you know that you want to share with the class, we would more than more than just welcome it. So please do share if at all there's anything you want to share with the class. Anyone? I think we can have that at the end of the class. If anyone wants to share, surely we can do that. Okay, so
Sure,
we can, yeah, so we'll move on with the chapter. Okay, so now mutations, like we we're just talking about mutations. When we talk about it, is, yes, abnormal cell division, also your DNA sequence. Okay, it could have mutations, but mutations don't happen rampantly, or they don't keep recurring. Mutations are something that is a rare happening, and one of the consequence of mutation, all of us know, is cancer or certain damage that is happening to your body, right? Mutants, or mutagens is what we call substances or chemicals that could lead to mutation. Can anyone guess what could lead to mutations? It is something that is a basic knowledge, and all of us know there are certain chemicals that, or exposure to something that could cause mutation in our body. Yes, absolutely. Radiation. What else? Carcinogenic particles. For example, what could be carcinogenic to us? Tobacco. Yes, absolutely. Yes, it is. Yes. Also, people who have certain, absolutely. Even people in certain occupations, that's why it's called occupational hazards, right? People who even work in certain places where there is exposure to fumes continuously, people who work in the mines. Yes, absolutely. So all of these are, even hair dyes for that matter, these simple chemicals that are there even our day-to-day lives, they also are, to an extent, they can initiate the carcinogenic process. But does it always happen? No, it doesn't. These are mutagens that have the possibility of causing mutations in our body. And a very good example is, even doctors, they do not tell you to get scans done every now and then, right? You do not get it done because anyway you're exposing yourself to radiation. So you don't, just like a blood test, you don't go for an X-ray every month, right? So this is usually why we need to make sure that whatever we can control around us, whatever factors are under our control, we make sure that we keep them away, especially when we know that they are mutagenic. Okay, now coming back to mutations. Mutations is nothing but a phenomena that causes an unwanted, or a sporadic change in your DNA sequence. Now, when a mutation happens in that stretch of your DNA, that does not, let's say it does not produce any protein, because like I told you, not all genes produce protein. So if there is a mutation happening in a part of your DNA where there is literally no function, it's not going to cause much change, right? It's not going to cause any difference to your body, not on such a grand level. But let's say there is a mutation happening at the promoter region. What's the promoter region? Is where a particular, there could be an initiation of making a protein. So usually when a mutation happens in a part of DNA or a stretch of DNA that has a good function. Let's say there is a stretch of your DNA that's responsible to suppress a tumor. So if there's a mutation that happens in that area, don't you think it is going to lead to a particular consequence that is going to be more grave for your body? So mutations do not happen very rampantly. They happen rarely. But there is one type of mutation called single nucleotide polymorphisms, or SNPs, which are very common. Okay, SNPs, or those tiny mutations, could be responsible for you metabolizing a particular nutrient in a certain way and me doing it in a different way. Even for identical twins, if their DNA would be completely similar, of course, they are identical twins, so the genetic material will be the same. But the SNP, or the single nucleotide polymorphism, which means, what is a nucleotide? Do you remember seeing adenine, thymine, cytosine, guanine that we saw, the nucleotide? So if there is one nucleotide that is replaced by something else, that is called a nucleotide polymorphism, which means these can be there in, throughout your DNA. It could be at various places. It may not cause such a great health issue, but it will be a variation. So these variations happen even in identical twins. So even if they have the same genetic material, their SNPs will be responsible for one twin probably becoming obese at some point and the other still staying skinny. Okay, so this is also a debate when it goes on in nature versus nurture. You're born with your DNA, which is your nature, but your nurture is what you expose yourself to. Your surroundings constitute your nurture. Okay, so now genetics, this is what you've already seen. Genetics, what is the study? It's a study of course, heredity, but it's also variation, because heredity or evolution we have studied has given rise to variation. Variation in people that belong to the same family, the same community, or different communities, even in a particular population or two populations from different areas. Okay, so variations help us in understanding human genetics. So it's the basics of human genetics, which gives rise to evolution, right? We have a common ancestry, but then we still are very different in our adaptation. Someone mentioned adaptation, right? So all of this comes from now. Causes of evolution. Why exactly evolution could happen? Because environment is consistently changing, constantly changing. Now imagine thousands of years ago, hundreds of years ago, when we think about the situation or the environment, the kind of living or the lifestyle that human beings had, and now how we had, there is a vast difference. You can't even imagine the difference, right? Because as a species, we are evolving, we are progressing. We are making sure that there are so many medicines, there is science, developing, because as a species, we are moving forward. And what we need to make sure that we conserve our species and bring it to an optimal level of health and lifestyle. So when it comes to evolution, that is one of the reasons. Also genetic drift. Now, when we talk about calamities, natural calamities that is not under our control, maybe volcanic eruptions, droughts, hurricanes. So a population that undergoes that, they will move to a different location and starts from scratch. That also leads to adaptation within ourselves, that leads to variation. So the next generation that comes after that, and next to next generation that comes after that, they will show much variation than what their ancestors showed, right? If you compare yourself with maybe a tribal area that's living in a drought, they don't have water for 6 months in a row, but they have adapted in a certain way. If we put ourselves in that place for a week, we won't be able to survive. But they have adapted, they have changed. That is the variation they show, right? So now that is because of genetic drift. And what is mutations? So these are mistakes. right? Mutations is not something that we intend to happen to our body, or something that, you know, theek hai, abhi pandemic hai, there's this virus that is, you know, very deadly. So if I tell my body that, you know, you need to mutate in order to adapt to that, it's not going to do it, because mutations do not happen as per what we intend. They can actually happen as and when, you know, there is an exposure to mutagens, or it could be sporadic inside your body. So at that stage, so that could be more deadly. Gene pool, genetic biodiversity, and allele frequency are all ways of understanding the evolution. Okay, so usually what happens is understanding the property of mutation. Like I said, what is allele frequency? A particular trait and the frequency of that particular trait in a gene pool. A gene pool is, let's just say we are taking a population. Okay, so one particular allele or a trait is usually counted in a particular population, and it is divided by the number of alleles. So this is how frequent this particular allele or this particular trait is. But why is this important? Again, understanding evolution, understanding variations, and also understanding mutations. Because what happens is, if a particular mutation is frequenting in a particular population, it is not considered as a mutation, it's considered as a SNP or an SNP, which is more common. If it is less than 1%, it is considered as a mutation, because mutation is not common. It happens, but it does not happen as, like, you know, it is rampant in a particular population. Okay, so this is mutation. So there are different types of mutations. Let's understand them first. So a silent mutation, like the word suggests, is something that's not going to cause much of a change. Now let's just take an example of codon. Okay. Now if there is a particular amino acid that is being coded by, you know, one codon, let's just say this codon, one of the nucleotide is replaced by another, but the other codon is coding for the same amino acid. We saw earlier on, remember, that there could be multiple codons coding for the same amino acid. So now let's just say that there is a mutation in one particular codon, wherein one of, let's say, U is again replaced by something else, but the codon is coding for the same amino acid. Let's just say serine is an amino acid that it's coding for. In this case, what it will code for in the end is going to be the same thing. So even if there is a mutation, there is a nucleotide replaced by something else, it's finally coding for the same amino acid. So it's not going to cause any mutation that is detrimental to the person. It's still going to cause the production of the same protein, which is why it's called a silent mutation. It does not cause any detrimental effects. Point mutation is nothing but, it's just like a silent mutation, but it's a single amino acid or a short sequence is substituted for another one. But point mutations, depending on where the mutation is occurring and for which amino acid, it can cause a change. It could either be harmful or beneficial. Missense mutation could actually change the code for the entire protein. What if it changes the code? An entirely different protein could be made. So this can cause an issue. It is missense mutation, meaning you cannot make sense of the protein that is actually formed in the end. Nonsense mutation is nothing but it codes for a stop codon, where, let's just say, a protein is made, right? The elongation process is happening. Now suddenly there comes a codon right in between which codes for a stop codon before the protein is complete. So this is a nonsense mutation, because of which the mutation codes for a stop codon in this case. And SNPs or SNPs, it's pronounced as a SNP, but it's called SNPs. So single nucleotide polymorphism is the most common type of genetic variation amongst people, even identical twins, like I mentioned. So each SNP represents a difference in single DNA base. For example, let's just say nucleotide cytosine is replaced by thymine in certain structure of DNA. That could either give you protection towards a particular health issue, or could actually render you at a risk of that particular thing. Okay, so these are SNPs, which is the reason why we respond to different things differently. despite having just 0.1% difference in genetics. So mutations could also be classified into different ways: deletions, inversions, duplications, or substitutions. Okay, so now deletions of vital segments means there is a deletion of an entire thing. It's just like, you know what, you are editing a particular statement, let's say. Now you know that every amino acid is represented by a code of 3, right? If there is a deletion of an entire segment from that stretch of DNA, it's obvious it's not going to give you the right protein made in the end. It is going to cause either the protein to be mutated or it to be rendered useless. So that is deletion of a particular segment from a stretch of DNA. Inversion is when one segment is exchanged with the consecutive one. Okay, and duplication or triplication is when it is, of course, it's duplicated, but again, it is going to cause a change in the final DNA. Substitution, insertion, deletion, and frame shift. Let's see each one of these. Substitution, like the word suggests, is when there is a substitution of a nucleotide for another. Okay, so such a substitution could either result in the change of codon, which encodes a different amino acids, and consequentially it will lead to a different protein. So sickle cell anemia, if you know what that is, your cells, RBCs, they look like a sickle. They are not rounded and healthy. So they look like a sickle shape. Okay, so this usually happens due to a substitution mutation in your genetics. Okay, insertion, like the word suggests, is inserting. Now let's just look at this particular line. Okay, now you will read only in a set of 3, right? Now if there is an insertion here, don't you think that this entire this entire segment is going to be divided unequally? So after reading the first section, it's going to take the next 2. But if there is an insertion of a different codon, the entire meaning changes. Similarly, deletion, there is a deleting of a segment. So only the first 3 will be read first, and then you have a single one, and then the next consecutive codes that are written on the side. So again, deletion also leads to something else. The frame shift is very interesting, and it has been used to explain to you using this particular statement. Now the fat cat sat makes sense, doesn't it? Let's just say there is a deletion that happens. Frame shift can happen because of any of these. So in this case, if let's just say T is deleted, what is going to be the result? Since it reads the first 3 and then the next 3 consecutively, it reads as HEF, ATC, ATS, AT. But does this make sense? To us, it's a statement that does not make sense. But in case of genetic level pay, they won't understand, it won't even code for an amino acid that it is responsible for. So this is frame shift mutation, which again codes for an incorrect protein. So all of these could lead to mutations, but like we said, it's not common. It happens sporadically. It happens only on exposure to certain factors or certain places where DNA replication or repair is concerned. But again, you all don't have to worry that it will happen in these stages, because your body is very smart. It regulates on its own. It makes sure that it bypasses all of these particular hindrances. That's the reason why mutations are rare, right? So that's because your body has its own regulatory mechanism. Just like every organization has its own regulatory system in place, your body has its own regulatory system in place. Okay, so again, it was not a very long chapter, but it's very interesting. All right. Now, the next chapter, which is genetics in healthcare management, it is not technical. It is something for a read up only. It is not going to involve any, technical aspects or fundas in genetics. It is just about how it helps in healthcare. Okay. Now, this we already know. We don't have to go into it, one by one. Now, let's just hear from all of you. What exactly do you think genetics could have a role in healthcare management? We know that it is a predictive and a preventive tool, right? So now these are the ways by which genetics in healthcare holds a particular place. Okay, gone are the days where, of course, once the symptoms start, we actually get to know that we have a particular health issue. But genetics not only helps in prognosis or understanding what could be the risks that you carry, but it also helps in forming, or it helps in forming the basis of diagnosis. Even for physicians, for doctors, they have now used or integrated this particular tool in order to make their diagnosis, the treatment, the targeted treatment better than what it used to be. So genetics in healthcare management can be used for various reasons. Diagnosis, it is not used in diagnosis as such. So don't get it wrong that, a genetic report is going to help you diagnose something. No, it helps in diagnosis in the cases where, a person has a certain recessive linked disorder or a dominant linked disorder where it has come from the family. In that case, it helps in diagnosis. But otherwise, when you talk about wellness segment, wherein we understand your risks, so that can help side by side, integrated with the diagnostic tools, it can help in diagnosis. Okay, prognosis, of course, in understanding the predictive nature of genetic reports. Prevention. Now, if I am told that you will get to understand your risk of diabetes and heart disease, I will ask you, why should I get it done just to know my, just to predict? I mean, it's like I'm going to keep thinking about it until I actually end up with that health issue. So why exactly is it important? Because it's not only going to tell you, what you're going to end up, there is a chance that you'll end up with diabetes, but it also helps you in understanding how to prevent it. So sometimes the solution to your issues or your health issues that you could be prone to lie in your genetics itself. When you see the report that we have attached for you in this particular, in this course, towards the end, in the last, affecting the moment it is born. Okay, so this is the reason why carrier screening is done. And if you remember, we did when we studied the different types or the subtypes of genetics, the genetic disease that is being carried. Somatic gene therapy obviously could happen, once a person has acquired certain disease. In order to treat it externally, a DNA is introduced. So let's see how exactly that is done. First is gene augmentation therapy. What does augmentation mean? Augmentation means introducing or, integrating something. So when it comes to a cell, let's just say it has a non-functioning gene. If it's non-functioning, the gene is not going to perform the function that it needs to or won't produce the protein. So it is useless. So now you put a functioning gene into the cell. This is called gene augmentation, where if there is a non-functioning gene, there is externally introduced a functioning gene that can help the cell function normally. Now, gene inhibition means what? Let's just say a cell contains a faulty gene because it's producing either a protein that's not needed or it's producing some toxic material for your body. So now there is a blocking gene that is introduced in this particular cell. Now, unlike the previous one where there was a non-functioning gene where you introduced a functioning gene, in this case, you put a blocking gene. Why a blocking gene? Because this particular cell's faulty gene is going to produce a product. But now this blocking gene will block the faulty gene's new product. Okay. Now, when that happens, your cell will start functioning normally. Okay. And the other way is killing of specific cells. Let's say a cell is diseased. If a cell is diseased, it's of course going to cause issues inside your body. So now there are two ways by which this is achieved. One is there is an introduction or an insertion of suicide gene. Now, like the word suggests, what do you think it's going to do? It's going to produce a toxic product. This cell is already diseased. It's of no use, but then it is going to be detrimental to your health. So in order to kill this cell entirely, there is a toxic product that's produced by the suicide gene. And just like that, the diseased cell is going to disintegrate or it's going to die. But there's another way by which this is achieved. Similarly, there is a diseased cell. Now there is a marker gene that is introduced. Do you know what a marker gene does? Your cells, they have certain receptors on top. Okay. Now these receptors could attract your body's own immune cells, and that could attack them. Okay. Now, usually when you have these viruses that enter your body, they have certain receptors that initiate an immune response from your body. Okay, that happens almost to every foreign body where your body recognizes it as foreign and it starts attacking it. Now, in this case, we use that mechanism of our body in order to direct the killing of that particular cell. Now, once we introduce the marker gene, what's going to happen? The marker proteins are going to show up on top of that cell. Because of that, your immune system is going to attack this particular cell, and that is going to cause the disruption of the disease cell. Okay, so now these are the ways by which gene therapy is done. Okay, so how exactly is the DNA transfer done? A very common or a simple example that I would give you and you would relate to it is, the vaccines. It is a virus vector vaccine. Certain vaccines are virus vector vaccines. What do you understand by virus vector? Vector is nothing but a vehicle that can be used in order to generate a response in your body. But now, what happens when a gene therapy is done is, of course, a virus vector is used to introduce that gene into a cell. But there are always drawbacks to this. Okay, this is usually done, but there have been certain drawbacks when it comes to this gene therapy. So the challenges were delivering the gene to the right place and switching it on. Gene to deliver kar diya, but then usko switch on bhi karna hai for it to generate a response. Also, delivering it to the right place is important. Otherwise, it can interfere in the action of other genes as well, right? Avoiding the immune response. Now see, we are using a vector or a vehicle, but regardless, your body is going to identify it, okay, as something that it needs to attack. So how to avoid that immune response? There will be some amount of immune response, but not such an immune response ki matlab wo gene that is supposed to help you gets destroyed in the way before it reaches the place where it's supposed to be targeted. Making sure the new gene does not disrupt the function of other genes. What will happen in that case? If there is another gene that's producing a protein, if this comes in the way of that, it's going to get nullified. You do not actually get a good response from either of the. So that could cause the new gene to come in the way of another gene that could disrupt its activity. Okay, and the cost of gene therapy, of course. Now, cost of gene therapy is high because, of course, it's not done on a commercial level. It's not done, like I said, you go into a clinic and you get it done on you. Gene therapy is very expensive, and usually, or for that matter, when you have gene-based therapy in which a particular medicine is targeted towards a particular genetic health issue, it is not changing your DNA sequence on any level. What genome editing does is it actually changes your DNA sequence. That is why it's called editing. Okay, so now genome editing is a technique that is precisely and effectively done to modify the DNA within a cell. Now, it could be done for various reasons, right? If a person is diseased, if a person already carries a genetic disorder where the person wants to get rid of that particular gene responsible for generating that response in the future. So genome editing can be done for various reasons, and it has been applied, it has been done. And there is one technology that there was a Nobel Prize winner for that in 2020. We saw it in the first chapter. Who remembers what is the name of that genome editing? What is the name of that editing technology? Can anyone recall? There are many editing techniques, but this is one technique that received the Nobel Prize in 2020 recently, with two women scientists who actually introduced that technique. Does anyone remember? Come on, it starts with a C. I've given it away. No one, I'm sure some of you know, you'll have already gone through these chapters. Okay. The name of the technology is CRISPR-Cas9. Okay. Now, CRISPR-Cas9 is a genome editing technique that won a Nobel Prize for its work in research, okay, in order to treat certain diseases. Now, like I said, genome editing is not done usually for everybody. It's not like a treatment method, but it is used for research to treat certain diseases that is still underway in understanding how to treat them from a genetic perspective, and for biotechnology, even in agriculture. You know, in order to make plants resistant to, let's say, pesticide, the plants can protect themselves, right? You don't have to spray anything on top of them. So they're genetically modified in order to, let's say, to even counteract drought-like conditions. Okay, so all of this is used in order to understand this particular technology. So genome editing is very, very, it's used, it's utilized, of course, it's underway. A lot of research has been done on it. So let's see what genome editing and how it exactly works. In all of the, for the same amino acid, let's just say serine is an amino acid that it's coding for. In this case, what it will code for in the end is going to be the same thing. So even if there is a mutation, there is a nucleotide replaced by something else, it's finally coding for the same amino acid. So it's not going to cause any mutation that is detrimental to the person. It's still going to cause the production of the same protein, which is why it's called a silent mutation. It does not cause any detrimental effects. Point mutation is nothing but, it's just like a silent mutation, but it's a single amino acid or a short sequence is substituted for another one. But point mutations, depending on where the mutation is occurring and for which amino acid, it can cause a change. It could either be harmful or beneficial. Missense mutation could actually change the code for the entire protein. What if it changes the code? An entirely different protein could be made. So this can cause an issue. It is missense mutation, meaning you cannot make sense of the protein that is actually formed in the end. Nonsense mutation is nothing but it codes for a stop codon, where let's just say a protein is made, right? The elongation process is happening. Now suddenly there comes a codon right in between which codes for a stop codon before the protein is complete. So this is a nonsense mutation because of which the mutation codes for a stop codon in this case. And SNPs or SNPs, it's pronounced as a SNP, but it's called SNPs. So single nucleotide polymorphism is the most common type of genetic variation amongst people, even identical twins, like I mentioned. So each SNP represents a difference in single DNA base. For example, let's just say nucleotide cytosine is replaced by thymine in certain structure of DNA. That could either give you protection towards a particular health issue or could actually render you at a risk of that particular thing. Okay, so these are SNPs, which is the reason why we respond to different things differently, despite having just 0.1% difference in genetics. So mutations could also be classified into different ways: deletions, inversions, duplications, or substitutions. Okay, so now deletions of vital segments means there is a deletion of an entire thing. It's just like, you know what, you are editing a particular it reads as HEF, ATC, ATS, AT. But does this make sense? To us, it's a statement that does not make sense. But in case of genetic level pay, they won't understand, it won't even code for an amino acid that it is responsible for. So this is frame shift mutation, which again codes for an incorrect protein. So all of these could lead to mutations, but like we said, it's not common. It happens sporadically. It happens only on exposure to certain factors or certain places where DNA replication or repair is that much. For example, thalassemia. Okay, it may not. It's not just telling you that this code regulatory system in place. Your body has its own regulatory system in place. Okay, so again, it was not a very long chapter, but it's very interesting. All right. Now, the next chapter, which is genetics in healthcare management, it is not technical. It is something for a read up only. It is not going to involve any technical aspects or fundas in genetics. It is just about how it helps in healthcare. Okay, now this we already know. We don't have to go into it, one by one. Now let's just hear from all of you. What exactly do you think genetics could have a role in healthcare management? We know that it is a predictive and a preventive tool, right? So now these are the ways by which genetics in healthcare holds a particular place. Okay, gone are the days where, of course, once the symptoms start, we actually get to know that we have a particular health issue. But genetics not only helps in prognosis or understanding what could be the risks that you carry, but it also helps in forming the basis of diagnosis. Even for physicians, for doctors, they have now used or integrated this particular tool in order to make their diagnosis, the treatment, the targeted treatment better than what it used to be. So genetics in healthcare management, when it builds up, it leads to mental retardation in these children later on, which is the reason why newborn screening is very, very important. Newborn screening can also be done for genetic linked disorders or even abnormalities that happen at birth. Carrier screening, like the word suggests, carrier, is the people who carry it, but it doesn't affect them as much. So in this case, we are talking about the parents that may carry a particular gene. It may not affect them that much. For example, thalassemia. Okay, it may not affect them as much, but if in case the child ends up having a double risk copy, the child will be affected the moment it is born. Okay, so this is the reason why carrier screening is done. And if you remember, we did when we studied the different types or the subtypes of genetics, there was this pre-implantation techniques. So that also can be used in integration with this particular kind of a therapy. Then you have gene therapy and gene-based therapy. These two are completely different. One is invasive and one isn't. Okay, so we'll see what exactly is the difference between the two. We have the last chapter which involves gene editing. So you will see whether that is gene-based or that is gene therapy. So what is gene-based therapy? Can you all guess? So is gene-based therapy going to change your genetics? Like, are they going to change your genes like manually? No, that is gene therapy, where your genetics is actually changed or modified. But this is not done like on a commercial level where, you go into a clinic, walk into a clinic and get gene therapy done. No, it is done. It is still it has been done. There have been a lot of cases where it is still undergo in order to understand how to cure certain diseases that till now have no cure. Okay, gene-based therapy, on the other hand, is on the basis of your genetics, how certain medications, certain drugs can be targeted or they can be devised in order to target your genetics. Okay, so this is gene-based therapy, and there are many drugs that have been made. So a person, if he's on chemotherapy on the basis of his genetics, an oncologist can actually decide which kind of chemotherapy will work better for that particular person. So this comes under gene-based therapy. Okay, let's just see what gene therapy is. That was gene-based. This is gene therapy. Now, gene therapy is when externally a DNA is introduced into a patient to treat a genetic disease. There are two types, somatic and germline, which we've seen these terms come so many times. So somatic, we already know when it is directed or targeted towards other cells of the body that are not reproductive in nature, while germline, the word germ itself means reproductive cells. So germline gene therapy is when a transferring of section of DNA is done towards the cells that are reproductive in nature. Okay, germline gene therapy usually helps in preventing the future generations from being impacted from this particular, from the genetic disease that is being carried. Somatic gene therapy obviously could happen, once a person has acquired certain disease. In order to treat it externally, a DNA is introduced. So let's see how exactly that is done. First is gene augmentation. Therapy. What does augmentation mean? Augmentation means introducing or integrating something. So when it comes to a cell, let's just say it has a non-functioning gene. If it's non-functioning, the gene is not going to perform the function that it needs to or won't produce the protein. So it is useless. So now you put a functioning gene into the cell. This is called gene augmentation, where if there is a non-functioning gene that is externally qualified, you do not actually get a good response from either of the. So that could cause the new gene to come in the way of another gene that could disrupt its activity. Okay. And the cost of gene therapy, of course. And been done on it. So let's see what genome editing and how it exactly works. In all of the genome editing, okay, there are two parts. There is one part that cuts the DNA and the other that directs the scissor towards the section of DNA. Now, scissor to hai cut karne ke liye, but how does that particular scissor know which place to go and cut? It can go and cut any side of the DNA, right? So it needs someone to guide it. So there are two parts. One is the guide that helps the scissor, and the other is the scissor that actually cuts the DNA where the editing needs to happen. Okay, so this is how a genome editing is done, where there is an enzyme called engineered nuclease. So this nuclease, like the word suggests, nuclease is the one that cuts because it's an enzyme. Okay, enzyme, breaks down. So in this case, remember, it's an engineered nuclease that helps in cutting the DNA. But there is a DNA targeting part also. It could be an RNA or something else. We will see in the next, paragraphs. So an engineered nuclease and a guiding part. These two constitute genome editing in a nutshell. So what are the types of genome editing? This is simple for you to understand if you understand how normal editing is also done. So one is small DNA changes. Let's say you want to cut a certain part of DNA. Okay, now there is a particular small section of DNA which you do not want. So you make a cut here. So a small DNA change is included in that. Removal of a section of DNA. So there is an entire section that needs to be cut. And the third is insertion of a section of DNA. Now, how would you insert unless you actually remove a particular segment? It's just like you have a statement. Now, I want to remove this sentence and I want to replace. When it comes to other systems, it's not that also involves same two things, wherein there is you have CRISPR-Cas9, which is, of course, now it's become very popular. So CRISPR-Cas9 is the most common. It's a cheap and efficient system for genome editing. So CRISPR stands for, it is a mouthful, but clustered regularly interspaced short palindromic repeats. Okay, now CRISPR is the one that targets the DNA, while Cas9, which is the other part, is the nuclease that cuts the DNA. So this is how it goes. Cas9 is your scissor, and then you have your guide RNA. Why is this guide? How does it exactly direct? Because it carries the complementary, the codes that are responsible to direct this scissor to that particular area. So it's going to guide it to that area where it is complementary to. Okay, and this is how there is edits made in that area. Now, this particular system is the most common. It is cheap and it can be used, on a larger scale in order to, it can be done simultaneously to understand different diseases. When it comes to other systems, it's not that it is not efficient, but it is a little complicated. It is more complex than what CRISPR-Cas9 is. So ZFNs is also known as zinc finger nucleases. It's made up of zinc finger proteins, wherein that helps in guiding. Okay, this is the one that guides, but there are two molecules for that. So two molecules side by side, they guide it to the fork nucleus is the cutting part. So these two guide on two different DNA strands, and then that particular strand is edited. And talons also works just like ZFNs. So it stands for transcription activator like effector nucleases. Don't go on the name. So talons also involves same two things, wherein there is a guiding RNA. At the same time, there is fork nuclease, fork one. So here it works in the same way like ZFNs, but regardless, these two are not that, easily used, or it can be used very effectively. So CRISPR-Cas9 takes the prize amongst all of your genome editing systems. But like any genetic editing system or any genetic research, there's always limitations to this as well, right? To improve the specificity, so it needs to be more specific. There can't be any mistakes. It needs to go exactly to that particular area where it needs to cut, and it needs to do it fast. You remember, like I told you, if in case there is an insertion required, but then before that the genome, your DNA starts repairing itself, it's not going to be that efficient. Understanding DNA recombination and repair machinery, the same thing. We need to understand it so that we can make it better and with less mistakes. Okay, CRISPR is better and cost efficient in comparison to others, that is your ZFN, talons, and there are some others as well. So it can be used for large scale research. Genome editing, again, it also occurs as a natural process in your body. Right now, you also know that there are viral agents or viruses. They are also RNA, right? So they also can cause certain edits in your system. So that also comes in the way. Human enhancement. So human enhancement is nothing but a goal basically for genome editing systems. So this is what it is done with a goal. How can human beings evolve and progress better?
And paste progress, otherwise you will be we'll have to rush up at the end, and then you will just do it for the heck of doing it. So and from now on you understand the pattern, so answer the questions and ensure that you all are doing you all are doing really well. Don't rush up and lose marks. I think you're doing phenomenally well. Kalpana Chauhan is doing phenomenally well at this point in time. Okay, so study hard. I'll give you an update next week as well.